Hi, so today we're going to be talking about cell respiration. Basically, it is a redox reaction that is similar to photosynthesis in that it is a metabolite gets oxidized and another metabolite gets reduced. So same thing as we saw in photosynthesis. The only thing is cell respiration, the whole process and the goal of cell respiration is to break down nutrient molecules that will be able to produce an energy source, and for us that's ATP. Essentially what you're able to do is utilize electron energy that's extracted from the breakdown of a glucose molecule, and that happens in some steps that we will definitely learn about. It's a really good way to make ATP very efficiently. And so that's all cell respiration is about. When you think about, okay, what is cell respiration? What is the whole purpose? And the whole purpose is generally to make ATP as efficiently as possible. This shows you cell respiration as an oxidation reduction reaction. So glucose gets oxidized. So remember, oxidation is losing oil rig. Remember your oil rig. Oxidation loses an electron. Reduction is gaining an electron. And so in this reaction for cell respiration, oxygen gets reduced, so you gain that hydrogen. Essentially, when we were learning about all of those important biomolecules, if you remember, ATP is one of those important biomolecules. It is adenosine triphosphate. And if you recall, there's an energy between the second and third phosphate because it's unstable. And so that bond, breaking that bond actually releases energy. And so when you have energy, you have the ability to do work. Some important components that we talk about in this chapter is electron carriers that are NAD and FAD. So those are very important electron carriers, and they carry that using a hydrogen molecule essentially. So FAD and NAD will carry hydrogens to the last step of the electron transport chain. So those very critical energy intermediaries, and not to say they don't act as ATP, but what they do is they pick up electrons along the way through the whole process of ATP synthesis, or the whole process of cell respiration. They pick up, at each step, they pick up electrons and they carry those to the last step of cell respiration, which is the electron transport chain. So with cell respiration, basically you're looking at glucose enters the bloodstream. We're able to digest that glucose. We process it. We convert that glucose into 2-pyruvate molecules, and we basically break that glucose down. Some of the important chemical abbreviations that we talk about for like glucose, oxygen, water, carbon dioxide, you need to know the abbreviations and the names. And there are other abbreviations that you don't have to memorize, similar to what we saw in photosynthesis. So cell respiration happens in 4 phases. There's glycolysis, there's the prep reaction, the citric acid cycle, and the electron transport chain. Basically, glycolysis is the breakdown of glucose into 2 molecules of pyruvate. So glycolysis happens in the cytoplasm, and it does not require oxygen to be successful. It does require, glycolysis is like, essentially the glycolysis itself means sugar splitting. And so to activate that glucose molecule, to break it down, you have to have a 2 ATP input. Essentially, you're able to make 4 ATP through this process, but you're only gaining 2 ATP because you actually required to start the process in the first place. So essentially, when we're learning about the steps of cell respiration, what I do want you to know is that my concern when I'm asking you about the process of cell respiration, I want to know where each step happens. I want to know how many ATP are made at each step, and what's the end result of each step. Essentially, in glycolysis, you were able to make 2 ATP, and you you broke down glucose into 2 molecules of pyruvates. And so both of those pyruvates are then used in the next step, which is the prep reaction. Some of your resources don't actually include the prep reaction as one of the pertinent steps of cell respiration. However, it is important. They say it's not really important because it doesn't really make ATP during this step, but it does become very important because the prep reaction takes those pyruvates that were created in glycolysis, that were left over at the end of glycolysis, and it prepares them by combining those pyruvates with a coenzyme A. And so then they enter the mitochondria as acetyl-CoA. And so essentially the prep reaction kind of prepares pyruvates for entry into the next step of the process. So then that takes us to the next step of the process, which is the citric acid cycle. So the citric acid cycle actually happens in the matrix of the mitochondria. So before we had glycolysis that happened outside the mitochondria, right? It happens in the cytoplasm. And then the prep reaction prepares those pyruvates for entry into the citric acid cycle. This happens in the matrix of the mitochondria. And in this process, you're able to produce 2 NADH, or you're able to produce NADH and FADH. And so, since it is a cycle, just like the Kaplan cycle that we saw in photosynthesis, the citric acid cycle also is kind of a similar process in that you break down that molecule of glucose. The citric acid cycle can turn twice then for each molecule of pyruvate that entered into the cycle attached to as acetyl-CoA. So for the citric acid cycle, you're able to produce 2 ATP per glucose molecule. And so what happens is you're able to produce NADH and FADH, which are electron carriers. They're going to carry electrons to the last step of this process, and you're going to be able to basically produce 2 ATP. So do your ATP math. I like to think of it as ATP math for cell respiration. So step 1, we made 2 ATP, and step 2, we didn't make any ATP, and step 3, we made 2 ATP. So 3 steps have gone by, and we've only really made 4 ATP molecules total. But if you're really paying attention, you're actually able to make 36 ATP per glucose molecule. So we're only up to 4 and we're up to step 3. There are 4 steps to this cycle, to this whole process. So if you can imagine, that 4th step is a very critical step in cell respiration. So the last step of cell respiration is the electron transport chain. And so basically the electron transport chain extracts energy from NADH and FADH, and it passes electrons from higher to lower energy states. And you're able to produce 32 to 34 molecules of ATP. I say 36 total per glucose molecule. So we mentioned earlier that NAD and FAD, or NADH and FADH, are big electron carriers. They carry electrons that are attached to hydrogen molecules. And so NAD picks up an H, FAD picks up an H, and carries them down the electron transport chain. So you're able to actually make a ton of ATP using this process. Essentially, I actually kind of like to consider I have a really cute analogy for NAD and NADH that kind of works for us. So when I picture this whole process, and this is an image of the whole process, you can see glycolysis happening in the cytoplasm. You can see that at glycolysis you made 2 ATP and 2 pyruvates, which were prepared in the prep reaction for entry into the citric acid cycle. And then the next step would be the electron transport chain and chemiosmosis, which we talk at greater lengths about a little bit down the road. But at every one of these processes, you can see that this process also sends electrons, see the little electrons, via NADH, those are carrying those electrons, and FADH, those are carrying, so NADH, 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 they're essentially carrying electrons to the electron transport chain here. And so what I imagine is that NADH and FADH are like little school buses, and they're busing little electron children, right? They're busing the little electron children to the electron transport chain school, okay? This is a big deal. So NADH, they're dropping their hydrogen / electron children at the electron transport chain school, and they're able to they're emptying their electron buses, they're able to go back and pick up more electrons and keep bringing those electron children to electron transport chain school. That's a big deal. So kind of a greater look into each of the steps of cell respiration. Glycolysis is the energy investment stage. You gotta use 2 ATP to activate glucose, so that way you get 2 ATP total out of it. So it's the energy harvesting step. And you essentially get 4 ATP max, but 2 of those were consumed to activate glucose. So you only make 2 ATP, and both of those molecules that you got from breaking down glucose, those G3Ps, get converted into pyruvates. This is a process and it looks really hairy, but if you really think about it, if you take a good hard look at this, it actually looks like the Calvin cycle that we saw in photosynthesis, but backwards. Because in the Calvin cycle, you're building that molecule. In this process, you are breaking it down. So you're seeing a lot of the same metabolites. And when we talk about substrate level ATP synthesis, it's literally just an enzyme taking ADP and a phosphate and bringing them together so that ATP can be made. It's very simple. So essentially, an interesting thing happens. So we said NADH and FADH take their little electron children and they drop them off at electron transport chain school, right? So that's great. However, like most schools, you can't have a school without a teacher. You can't drop off your children at school if their teacher isn't there and there's no sub. So at the electron transport chain school, oxygen is the electron transport chain teacher. Essentially, oxygen is the final electron acceptor in the electron transport chain that's embedded in the cristae of the mitochondria. So the FAD and NADH are dropping their hydrogens and / electron children off at electron transport chain school. Those electrons bounce along the electron transport chain until finally it's accepted by oxygen. And of course, hydrogens and oxygen make water, right? So cell respiration does make water at the end. If there's no oxygen available, there's no oxygen teacher at school, you can't drop off your electron children at school with nobody to teach them, right? So essentially, there's an alternative, and we call this an alternative, but we call the alternative as a sub. It's like a substitute teacher. So fermentation happens in the cytoplasm, and it is an option that's out there. It's anaerobic, so you don't need oxygen. So even if oxygen isn't there, the process can still work. The glucose gets kind of incompletely metabolized in this process, and unfortunately you make lactate or alcohol, which are both toxic for the cell. So if oxygen is available, if there's no oxygen teacher at electron transport chain school, then fermentation can be a doable substitute teacher. And what's great about fermentation is that you're able to drop off those hydrogen children with fermentation, or electron children, if you will. The electron is attached to the hydrogen. So you drop off your electron children with fermentation, the sub, and you can go back and get more electron children. So you can eventually take them to electron transport chain school when oxygen is back. So essentially, fermentation is an anaerobic process, and it doesn't require oxygen at all to happen. And what happens is NADH transfers its electron to pyruvate instead of to the electron transport chain where glucose can be completely metabolized. And so in yeast, that's how alcoholic fermentation happens. In bacteria and fungi, that's how lactic acid fermentation happens. So this shows you fermentation. You don't have to memorize this, but what you can see is NADH getting to drop off its electron children, and so it can go back and pick up more electron children. And of course, fermentation helps to produce lots of foods that we know and love. Cheese, more cheese, and the other stuff as well. Fermentation is advantageous because it provides you like a quick little burst of ATP for muscular activity, and it allows you to free up the NADH and FADH so they can go back and pick up more electron children. But it's disadvantageous because, of course, you produce lactate and alcohol, and that's toxic to cells. And it's not as efficient. You only get two ATP per glucose molecule versus 36 when you can completely break it down when oxygen is there. So then if oxygen is available, then those pyruvates can enter the can be completed by the prep reaction. So they get modified essentially by attaching to a coenzyme A to form the molecule acetyl-CoA. And acetyl-CoA can then enter the Krebs cycle. So the Krebs cycle, or the citric acid cycle, I call it the Krebs cycle because I like to give credit where credit is due, Hans Krebs. So it happens in the matrix of the mitochondria, so the center and gooey part of the mitochondria, and it begins by adding that 2-carbon acetyl group from acetyl-CoA to a 4-carbon molecule, which is oxaloacetate, and you actually form a 6-carbon molecule, which is known as citric acid. That's why it's called the citric acid cycle. In the citric acid cycle, you're able to utilize NAD and FAD to pick up electrons. They get reduced, so they gain hydrogens and electrons, essentially, so they can take those to the electron transport chain. You do make ATP, and you actually turn twice for one glucose molecule, or it makes sense, once for each pyruvate molecule. So you're able to produce 2 ATP. It happens in the matrix of the mitochondria, and you're also able to make some NADH and FADH. So this shows you the citric acid cycle. So the last step is the electron transport chain, and this happens for eukaryotes in the cristae of the mitochondria. In aerobic prokaryotes, it happens in the plasma membrane. And so what happens is, and this is not so different than the electron transport chain that we already learned about, where you're passing high energy electrons down carrier molecules to a final electron acceptor. So essentially, it's going from high energy, doing down the electron transport chain to the final electron acceptor, which is oxygen. And you're able to, during this process, those electrons are released from NADH and FADH, and they're able to go back and get more electron children for electron transport chain school. So essentially, electrons travel across the membrane, they're attracted to proteins and lose energy. And so that energy is used to pump hydrogens from the matrix into the inner membrane space, and that's called chemiosmosis. Those hydrogens actually then build up to high concentration, and those molecules are going to want to go from high concentration to low concentration. And so they are going to go right out that ATP synthase enzyme that we learned about, the same thing, same one, same kind of thing that we learned about in photosynthesis. And so what happens is, of course, ATP is formed by oxidative phosphorylation. And then remember, oxygen is the final electron acceptor, and it will combine with those hydrogens to form water. This is a nice setup of the organization of the function of the electron transport chain. You can actually see those proteins embedded in that, the cristae of the mitochondria. You can see those NADH, those little school buses dropping off their electron children to the electron transport chain school. And of course, you can see it's very important that they do, so they can be recycled back into the system. So it's super important that you have those NADH molecules and the FADH molecules that can go and drop off those hydrogens and electrons, and they can actually go back into the system and pick up more hydrogens and electrons, right? So we saw when oxygen isn't available to receive those electron children, that NADH and FAD can't free up. So it's kind of a big deal. Energetically speaking, when you can fully break down glucose, you get 36 ATP total. And that is, it's 39% efficient, which doesn't seem great, but we're not photosynthesizers. We're not as good as them. So now we know that the mitochondria, well, it used to be, we used to say it was the powerhouse of the cell. Now it's where ATP synthesis occurs via oxidative phosphorylation.